medical case and using it to look at things like action potentials and seeing how it influences things as simple as resting potential. It's kind of esoteric until you see it used in an actual case study. So this is another case where you can really see the need for knowing the underlying physiology when you're looking at synaptic kinetics in the mind. There are lots of conditions that can block neurotransmitters and give you trouble then. So knowing the normal physiology is important so that you can understand if you have any pathologies going on. So synaptic transmission, new chapter 13. The history of it is, I think, really interesting. How did you figure out that you have a chemical diffusing across the synapse? At first it was thought, we use this electrical spark. It came up the nervous system in electrical terms. And there was this spark versus the soup. Is it a chemical, the soupy mixture going across the synapse? So it was the spark versus the soup. And you had to pick which one. And the early studies were trying to elucidate that, to figure out, is it electrical or is it chemical? So Elliott, 1904, had one of the very first instances of chemical being isolated. So the adrenal gland was able to grind that up and extract epinephrine from the adrenal gland. So known as adrenaline. And then the study by Lowey in 1921. This is a really awesome study. And this showed that, yes, cell-cell communication is chemical. What he did is put, if you remember our heart experiment from Bioconcepts 2, he put a heart in a solution and zapped it. And when you zap it, especially the vagus nerve, it releases acetylcholine. Didn't know that there was a chemical release, but if you zap that vagus nerve, it causes the heart rate to slow down. He took the solution that heart was bathing in and dripped it on heart number 2, pumped it over so that only the saline that it was bathing in was touching heart number 2. Heart number 2 slowed down. Didn't have to zap it. So it must be some chemical substance that's responsible for causing this reduction in heart rate. So that's how he was able to experimentally determine that neurotransmitters are used in cell-cell communication. So, very cool thing. And that transmission between the vagus nerve and the heart, they were able to later then isolate and say, yep, it's acetylcholine that does it. So, in our muscle studies in lab, it's kind of referred to, but we didn't really study the nitty-gritty details of muscle contraction. But acetylcholine in a muscle is excitatory. It causes your muscles to contract. Acetylcholine in the heart causes it to slow down. That's something that we'll see. You can modulate the response of nerves by having the same neurotransmitter but a different action. It all has to do with the receptors. We'll see why the receptors are super important as we go along. So this is the setup. Just a little terminology. The presynaptic cell is a cell that will be communicating, and we'll have the action potential coming down. So our presynaptic cell is up top, and then the postsynaptic cell is across on the other side of your synapse. So your postsynaptic cell, the presynaptic cell has all this neurotransmitter already stored up. So it doesn't have to manufacture it and then send it. It's stored up in these vesicles, just waiting to be released, diffuses across, and talks to the postsynaptic cell. So presynapse and postsynapse are what we're looking at anatomically. So we'll kind of trace the pathway to see what's happening in the presynaptic cell, what happens during the diffusion step, and then what happens in the postsynaptic cell. The structure of a chemical synapse can be seen when you're doing a scanning electron micrograph with some of these kind of artificially colored, so you can see all those vesicles, you can tell which cell is the presynaptic one, right? You have all kinds of vesicles being stored in the presynaptic cell, and then the postsynaptic cell is waiting to receive that message, and this little red line is the synapse. So the synapse is about 20 nanometers in width in a chemical synapse. So small, but significant. So you have your presynaptic cell, here's your vesicles, and your postsynaptic cell, the dendrites, often have these little buttons or bumps that will stick out, these little spikes and processes that will almost touch the presynaptic cell. So that's your synapse right there, where all that good physiology is happening. So if you want to just memorize this step by step, it would give you all you need to know about what's going on in the synapse. We're going to kind of go through and dissect this out and look at pieces of this to figure out how researchers came up with these steps. So, number one, first, your action potential will invade the terminal. This just means that it's coming down your presynaptic cell, coming down your axon, and it gets to the end, the terminal. So this is presynapse. Presynaptic cell. The action potential invades the terminal of the presynaptic cell. This causes gates to open, so calcium enters. Calcium enters the presynaptic cell, rushes in, and if it doesn't directly bind to vesicles, it binds to cytoskeletal elements that bind to your vesicles. So calcium comes in and interacts with the vesicles. This causes your vesicles that are preformed, already full of neurotransmitter, to fuse with your postsynaptic, or the membrane of the presynaptic cell. So the vesicles fuse and then release your neurotransmitter. NT is neurotransmitter. So the vesicles fuse. Exocytosis pushes it out into the synapse. So just like an amoeba would exocytose items from its vesicles. Vesicles fuse, neurotransmitter release, exocytosis. That neurotransmitter then has to diffuse across the synapse. The gap between the cells that's around 20 nanometers wide. When it gets to the other side, it binds to that postsynaptic cell and it binds to specific receptors. The receptors aren't always, but they're usually channels, right? So that they let in ions. So they bind to a postsynaptic receptor, often it's just ion mediated, so when it's bound, it opens and then ions can come in or out. So sodium could come in, potassium could come in, potassium could go out, chloride could come in. Binds to a postsynaptic receptor. And that receptor is often an ion channel. Okay. So the acetylcholine is just the messenger. It doesn't enter the postsynaptic cell. It just binds to the receptor on the outside, opens an ion channel, and then ions can come in or out. If ions are coming in or out, they're carrying a charge. So positives are coming in, maybe negatives are coming in. If enough positives come in, you can get an excitatory postsynaptic potential, your EPSP. That's excitatory postsynaptic potential. If enough negative charge comes in, you might get an inhibitory postsynaptic potential. So EPSP is excitatory. Postsynaptic potential. So we'll go into more detail about each of these things. This is just your summary. And then you could also get this inhibitory postsynaptic potential. If there's enough of an excitatory postsynaptic potential, brings your cell to the threshold, what do you get? Action potential. Okay, you might get an action potential if you have enough excitatory input. Okay, your postsynaptic cell has sent an action potential. You don't want your neurotransmitter just hanging around in the synapse. You need to kind of shut it down. So the neurotransmitter can separate from its receptor. It might just be destroyed, could be enzymatically broken down, and sometimes it's recycled and taken out by the presynaptic cell and used again, so that it doesn't go to waste. In either case, you probably have an enzyme in the cleft like acetylcholinesterase. Takes care of acetylcholine. So this is the summary that most neurotransmitters would follow. Questions? Lots of info packed in there. Now we're going to go through and take a look at these kind of step by step to see how this got figured out. There are interesting things that you can find in, like regarding calcium, regarding the neurotransmitter, the timing, just where and when it all happens. Okay, so just a visual summary of it. Action potential comes in. Vesicles are already here, pre-made, ready to go. Action potential comes in, that lets calcium in, that binds to the vesicles, they come to the presynaptic edge, and they exocytose the neurotransmitter. It all diffuses across, binds to channels that are specific for a certain type of ion, in this case it's sodium. Sodium comes in, positive charge, enough sodium comes in, you get an EPSP, and you get an action potential. Sometimes you get these G proteins activated. That's a little bit different. We're not going to worry about the differences. We'll mainly look at the ion channels. So synaptic vesicle at rest, waiting to go. You already have your neurotransmitter made and waiting in the terminal. So why is the sodium coming in? That's a really big question. Why is the sodium coming in this picture? Check with a neighbor. See if you can figure out why is that sodium coming in. This is hard. I don't know if that helps. Yes.
is sodium coming in to those channels? Yeah, because we have a depolarization happening, right? Thumbs up all around, yes? Yeah, depolarization means that sodium rushes in. You don't see potassium coming out, but your action potential has come down. So the voltage sensitive gates for sodium will open, sodium will come in. That depolarization due to sodium will activate the calcium channels. They're also voltage sensitive. So because you have a depolarization, the calcium voltage sensitive gates open, calcium comes in. Calcium is also in your extracellular fluid, especially here around the synapse. So calcium comes in, binds to your vesicles, they move, exocytosis. So that depolarization is important. Your ion channels open, in this case they're specific for sodium, so sodium comes in, and then the transmitter is cleaned up so that you don't have lots of acetylcholine still here. If you didn't recycle it, what would happen to this cell? Sodium or acetylcholine is still in the synapse.
It would keep sending the signal.
Keep firing, keep sending. You keep sending in sodium, because those channels stay open. So you want to kind of clean it up so you don't have acetylcholine just lingering in the synapse if you're done with it. Okay. So this summarizes that whole process. Acetylcholine, so calcium comes in, vesicles move, bind, acetylcholine is just a good one to use. Your neurotransmitter will diffuse across, bind. In this case, your sodium comes in. If you have enough sodium, you get that EPSP, excitatory postsynaptic potential. That causes different channels, your depolarization channels for your action potential to open. It comes in, you get an AP. So this sodium is causing your EPSP, excitatory postsynaptic potential. You have enough of it, it's strong enough, you get your action potential. Okay, so enough of an EPSP, you hit threshold, action potential occurs. Different sets of channels, and then you break it down with something like acetylcholinesterase, some enzyme in the cleft that will like recycle your choline. So the role of calcium is really important. And it took a while to figure out what calcium was doing. Calcium is a pretty important modulator of a lot of different signals, really. So it's worth looking at it a little closer. And so calcium, and the role of calcium was figured out mainly because of the timing of the events in the synapse. That's how they figured it out. So your action potential arrives, and it takes a while before your neighboring cell will fire its own action potential. So there's a delay. Postsynaptic delay was measured, and maybe half a millisecond. Sounds like not much of a delay, but it's a significant time compared to an action potential. So the diffusion of the neurotransmitter across that tiny 20 nanometer space, the diffusion itself only should take about 0.05 milliseconds, right? So just a fraction of that is based on the diffusion. So what's taking so long? It's not the diffusion of that neurotransmitter. Other things, other steps must be taking more time. So we have to look at the rate limiting step here. So why the difference? Well, a couple of experiments were conducted to figure this out, why it took long. And so one thing to do was to remove calcium from the extracellular space. So if you have something that binds calcium and removes its availability, if you don't have calcium in your extracellular bath, extracellular fluids, you don't get acetylcholine release. So no neurotransmitters show up in your synapse. And if you will label your calcium with this luminescent dye to see, okay, where is it? It'll confirm the finding. Is it absent from the extracellular space? When your action potential comes in, what's calcium doing? It showed that calcium's coming into your cell. So it showed that calcium's necessary for neurotransmitter release. So you bind the calcium, you can see that it's gone into your presynaptic cell, but not your postsynaptic cell. And therefore, because of these features that they found, researchers figured out that calcium entry was responsible for helping vesicles move. And that was the rate limiting step, getting calcium to come in, bind, reorganize the microtubules, move, take your vesicle, move it, exocytosis. That all takes a lot of time. So that was adding up to most of your 0.5 milliseconds. The actual diffusion, not that long. So calcium's important, and it takes some time to organize your microtubules. Now, if we had been able to do our frog experiment with the nerve, you can take your frog nerve and you put it in a bath of like our extracellular fluid. It's physiological saline, linear solution. And you can mix up different fluids, some calcium and some calcium free, and zap your nerve, and you can see that it doesn't work as well when you don't have calcium. So that's a rare solution. Did a lot of experiments like that where he would substitute and remove different types of ions until he figured out what was in a good physiological state. So that was what was done here. So if you look at depolarization of the terminal and compare that with our postsynaptic voltage potential, the postsynaptic cell voltage difference is going to go up only if you have enough calcium to do the job. So if you have enough calcium around, then you'll get a response because neurotransmitter is released. So calcium helps organize the microtubules so that they can grab the vesicles, make them bind to the presynaptic cell membrane, and then exocytose. Little docking stations here. And so the vesicles kind of get recirculated. You have this pool of vesicles that you refill, move it back up to the Golgi apparatus and you refill, repackage, and then you're ready to go again. That happens over and over. So then trying to figure out what is and is not a neurotransmitter became a problem. More and more chemicals were being discovered that have physiological responses. So what can cause a physiological response? Lots of things. Glucose can cause a physiological response, but no one would call glucose a neurotransmitter. So you had to kind of come up with a definition. So neurotransmitters have to be present in the synapse. That makes sense. And you have to verify it chemically somehow that it's there. And a really great tool to use to figure out what chemical might be in that synapse is to take a look to see how it's affected by various drugs, agonists that would kind of facilitate the action, or antagonists that might block the action of your neurotransmitter. So that can help you kind of pinpoint your structure of your neurotransmitter, what it might be. And neurotransmitters kind of get lumped into these different classes of compound, whether or not they're excitatory. Do they mimic other compounds that are known? So looking at, good old organic chemistry and underlying structure can really help you in figuring out the physiological change that you're going to be predicting. So all of these ways
are used to help describe what this neurotransmitter is, what the underlying structure might be. And then organic chemistry, using R, so the great instrumentation to figure out even more detailed than we were gonna worry about. So by doing these things, neurotransmitter examples were figured out. A lot of them have little abbreviations. So acetylcholine, great example. Acetylcholine used in muscle contraction as well as nerve response. Epinephrine tends to be excitatory. Norepinephrine. GABA, that's an inhibitory one. Aminobutyric acid. I have it written down on another slide. Serotonin, dopamine. These are really well-known ones that have been well-known for a long time. Others that might not be on your number one list. Glutamine and glycine can act as neurotransmitters. Histamine, even. You think of histamine when you think of allergies, but it could act as a neurotransmitter. There are other peptides, it's protein fragments, papuans. They can all act as neurotransmitters and cause a neighboring cell to create its own action potential and then transmit information. By far the most common examples are on the first list in terms of your neurotransmitters. And some are excitatory, some are inhibitory, and some can be either. So that really depends on the receptors that you've got. If you have the right kind of receptor, then, oh, you are excitatory. If you have a different kind of receptor, you're gonna be inhibitory. So acetylcholine can act either way. So the neurotransmitters kind of fit into these general categories, and you can see how the chemical structure is a little similar. So one category might be hallucinogens. So serotonin fits into this category, and I'm not gonna ask you this on an exam, but this is just kind of fun facts. So serotonin has a rather similar structure to things like LSD, mescaline. And you may, if you've taken medicinal chem, you've probably learned a lot of this stuff already. Stimulants, dopamine, epinephrine, norepinephrine, other stimulants, cocaine, methamphetamine, things like that. Depressants, GABA, gamma-aminobutyric acid, and things like ethanol. It's a depressant. And then narcotics, like decathlon. Looks a lot like morphine or oxycontin, that sort of thing. So having an idea of what class of compound it belongs to can kind of help you predict what these neurotransmitters are going to do. Okay, so those are the basics of neurotransmitters. Now, there's something called the quantum theory of neurotransmitter release. So, quantum theory of neurotransmitter release. So it's really important to start out by defining what a quantum is. And I always go back to physics and to just the study of light. So what's the unusual thing about light? Light is a wave, right? We talk about light waves, but it's also what? Not just a wave, but a particle. Yeah. And that's the whole quantum theory, the fact that it's a particle. So quantum is a particle, a discrete unit, right? A packet maybe, a particle. So a quantum, thinking about quantum is a kind of a discrete unit. That really factors into the neurotransmitter release.
because researchers were noticing this little phenomenon happening in your postsynaptic cell. It's important that it's in the postsynaptic cell. They were seeing MEPs, which are defined as miniature end plate potentials. So MEPs are these little, you'd use a voltmeter, right? And you'd see changes in voltage, changes in potential difference across your membrane. Voltage. So these MEPs, these miniature end plate potentials, are potential differences across the membrane, these voltage differences that you see in your postsynaptic cell. But they're tiny. They're not even an EPSP really. I'm just excited for it small. I guess you could call it an EPSP, but not big enough to be effective at all. Tiny. So they're very small. And the ones that were measured were positive. Teeny positive changes. And they all seem to be the same size. You could get, and not only were they the same size, they were integer units. So you could get 1 MEP or 2 MEPs, but you couldn't get a MEP and a half. Right? They tend to be these nice whole integer units. You could get 1 MEP. One miniature end plate potential showing up in your postsynaptic cell. If you get maybe 2 MEP showing up, but getting like 1.5 MEPs, that was not a thing. So that really piqued people's curiosity. Why? Why are you only seeing these whole number miniature end plate potentials? So what causes a positive change in your postsynaptic cell? So calcium comes into the presynaptic cell, and neurotransmitter goes across